Hello students, in today's class we are going to learn about tryptophan operon. So here we have the structure of the tryptophan operon, and it is divided into the structural genes, that is tryptophan E, D, C, B and A. Upstream of the structural genes is present the regulatory region, which consists of promoter, operator and a leader gene sequence. We also have presence of a tryptophan R gene that encodes for a repressor protein within the regulatory region. Um, so the product of the structural genes, that is tryptophan E, D, C, B, and A, are required for converting the substrate, that is chorismate, into the end product, that is tryptophan. The first enzyme of uh, the pathway, that is anthranilate synthetase, it is made up of two subunits each of tryptophan E and tryptophan D gene product, and it is involved in catalyzing the first two steps of the pathway. Anthranilate synthetase is also an allosteric enzyme. It has binding site for the substrate, which is known as the active site, where the substrate chorismate and further anthranilate will bind, while it also has another site which is known as the allosteric site, where the uh, end product of the pathway, that is tryptophan, can bind. Binding of the tryptophan to the allosteric site changes the structure of the active site of the enzyme such that the substrates are unable to bind, and as a result the pathway is inhibited when we have enough amount of tryptophan present in the cell. Further, coming to the regulatory region, promoter is the sequence where RNA polymerase can bind and carry out transcription. uh, Operator is the sequence where the repressor protein will bind. Then we have the leader gene sequence, which plays an important role in controlling or deciding if the messenger RNA formed is going to be a longer messenger RNA, including the structural genes, or if the transcription is going to terminate immediately after the leader gene sequence synthesizing small messenger RNA. This is regulated by a mechanism which is known as attenuation, which is brought about by the leader gene sequence. Then the tryptophan repressor gene will encode a repressor protein. Repressor protein can bind at the operator sequence, inhibiting the transcription of the downstream genes. This slide discusses the points on the structure of the structural genes and their role, and about the regulatory region and their role in the next slide. So coming to the repressor protein. So the repressor protein is encoded by the tryptophan repressor gene, which is formed after transcription and translation process. In this case, the repressor protein is not active by itself, but it requires help from a co-repressor molecule. Co-repressor molecule here is a tryptophan molecule itself. So the both can, when present, they will form an active repressor co-repressor complex, which is now capable of binding to the operator sequence. When the active repressor co-repressor complex is bound at the operator, RNA polymerase that is present at the promoters is unable to proceed, and hence transcription of the downstream genes will not take place. Such operons which are normally in and on state, that is the expression will take place. However, it requires a repressor molecule to stop the expression, are known as repressible operons. Then in the next slide, we have the sequence of the leader messenger RNA encoded by the leader gene sequence. So the leader messenger RNA has about 162 nucleotides. It consists of four regions: region 1, 2, 3, and 4. Region 1 contains two adjacent or tandem tryptophan codons, which are important for a tryptophan sensitive mechanism that will determine if the sequence 3 and sequence 4 can base pair with each other, or sequence 3 and 2 can base pair with each other. The sequences of the four regions is such that region 1 and 2 are capable of base pairing with each other, forming a hairpin loop like structure. Similarly, region 2 can also base pair with region 3, forming a hairpin loop like structure. The base pair between region 2 and 3 is also known as an anti-terminator structure. Uh, Region 3 can alternatively also base pair with region 4, forming a hairpin loop like structure, which is known as an attenuator or a terminator structure. Since followed by region 4, we have a poly-use sequence. So a combination of a hairpin loop immediately followed by a poly-use sequence is recognized as a transcription termination signal. So looking at the attenuation mechanism of regulation, there are two conditions. The first condition is when there are high levels of tryptophan available in the cell. So in case of prokaryotes, transcription and translation process takes place simultaneously. So as soon as some amount of messenger RNA is synthesized, it is immediately bound by ribosomes and they will simultaneously begin with the translation process. Now, when there is enough amount of uh, tryptophan that is present in the cell, you will have uh, more concentration of amino acid transfer RNA tryptophan present in the cell. As a result, the ribosomes proceed swiftly through the two adjacent tryptophan codons present in region 1, and hence it is also occupying, the ribosomes are also occupying part of region 2. By that time, the rest of the messenger RNA consisting of 3 and 4 region is also synthesized. Now, since region 2 is partly occupied by ribosomes, hence 2 is unable to base pair with region 3 as it is synthesized, while 3 and 4 are free and hence they base pair with each other, forming a hairpin loop like structure which is immediately followed by the poly-U sequence. These structures are recognized as an attenuator or a transcription termination signal. Hence, the transcript releases or dissociates from the messenger RNA. The transcript dissociates from the DNA at this point, and hence the complex simply dissociates, and the transcription does not continues into the downstream structural genes. While in the second condition, that is when the tryptophan levels in the cell are low. So here again, the ribosomes will begin with the translation process. And since the availability of amino acid transfer RNA tryptophan is low, because of low amounts of tryptophan available in the cell, the ribosomes will take a pause in region 1 where they are waiting for the transfer RNA tryptophan to bind to the A site of the ribosome. As the ribosomes are pausing, you have synthesis of region 2 and then region 3. Now, as 2 is free here, it immediately base pairs with region 3, forming a hairpin loop-like structure. Continued transcription will result in formation of a region 4. So here you have a base pair between region 2 and 4, followed by a sequence that can base pair normally with the template. And then you have the poly-U sequence, that is synthesized. Since the hairpin loop and the polyuse sequence are not exactly adjacent to each other, as a result, transcription is not stopped in this case, and you will have formation of longer messenger RNAs, including the structural genes tryptophan E, D, C, B, and A. So that's it about tryptophan operon. Here we have some questions for your practice. So do try solving these questions. Thank you very much, and kindly like, share, and subscribe if you like the content.